In the diet, carbohydrates are present as complex polysaccharides, starch, glycogen, and to a minor extent, as disaccharides, sucrose and lactose. They are hydrolyzed to monosaccharide units in the gastrointestinal tract. Cooking makes the digestion process easier. The process of digestion starts in mouth by the salivary alpha amylase. However, the time available for digestion in the mouth is limited, because the gastric hydrochloric acid will inhibit the action of salivary amylase. In the pancreatic juice another alpha amylase is available which will hydrolyze the alpha-1, 4-glycosidic linkages randomly, so as to produce smaller subunits like maltose, isomaltose, dextrins and branched or unbranched oligosaccharides. Starch right-pointing arrow dextrins right-pointing arrow oligosaccharides right-pointing arrow maltose and isomaltose right-pointing arrow glucose. The cells of brush border of intestine contain the enzymes, sucrase, maltase, isomaltase and lactase. These enzyme activities are present as two different complexes, in the jejunum, a sucrose isomaltase complex and a beta-glycosidase complex. The sucrose isomaltase complex itself has two different catalytic sites. The sucrase maltase site will hydrolyze the alpha-1, 2 bond of sucrose and alpha-1, 4 bond of maltose. The second site has isomaltase maltase site, hydrolyzing alpha-1, 6 and alpha-1, 4 glycosidic bonds. The beta-glycosidase complex also has two catalytic activities, one hydrolyzing the beta-galactosidic bond of lactose, lactase, and the other a beta-glucosidase bond of glucosyl ceramide, a glycolipid. Among the intestinal disaccharidases, lactase activity is most limiting and it is an inducible enzyme. Disaccharidases hydrolyze the corresponding disaccharides into component monosaccharides which are then absorbed. Table 8.1. The clinical applications of lactose intolerance are given in box 8.1. Absorption of carbohydrates. Only monosaccharides are absorbed by the intestine. Absorption rate is maximum for galactose, moderate for glucose, and minimum for fructose. Absorption of glucose. Glucose has specific transporters, which are transmembrane proteins. Table 8.2 shows a summary of the glucose transporters. Cotransport from lumen to intestinal cell. This process is mediated by sodium-dependent glucose transporter 1, SGLUT1, Fig. 8.1. Absorption from intestinal lumen into intestinal cell is by cotransport mechanism, secondary active transport, Chapter 2. A membrane-bound carrier protein is involved, which carries glucose, along with sodium. This sodium is later expelled by the sodium pump with utilization of energy. So energy is needed indirectly, Chapter 2. The transporter in intestine is named as SGLUT1 and the transporter in the kidney is called SGLUT2. SGLUT1 is faulty in glucose-galactose malabsorption. The SGLUT2 is defective in congenital renal glycosuria. Another uniport system releases glucose into blood. The same intestinal epithelial cells have a different transport mechanism on the membrane-facing capillaries. Fig. 8.2. Intestinal cells release glucose into bloodstream by the carrier mechanism called glucose transporter type 2, GLUT2. This transporter is not dependent on sodium. It is a uniport, facilitated diffusion system. Glucose binds to the transporter on one side of the membrane. When fixed, the complex changes configuration. This leads to the closure of the first binding site. At the same time, the binding site is now exposed on the inner side of the membrane, releasing the glucose. The process is called ping-pong mechanism. The GLUT2, facilitated transport, is involved in the transport of glucose across membranes, from intestinal epithelial cells to blood, Fig. 8.5, and from blood to cells like liver cells, beta cells of pancreas and kidney. GLUT2 is present in intestinal epithelial cells, liver cells, beta cells of pancreas and kidney. Since GLUT2 has a high Km for glucose, its presence in beta cells is ideally suited for sensing a high glucose level and releasing insulin, Chapter 9. So this mechanism enables the pancreas to monitor the glucose level and adjust the rate of insulin secretion. Actions of SGLUT and GLUT2 are shown in Figure 8.3. Glucose transporter 4. The glucose transporter type 4, GLUT4, is the major glucose transporter in cardiac muscle, skeletal muscle and adipose tissue, Fig. 8.4. GLUT4 is under the control of insulin. But other glucose transporters are not under the control of insulin. The clinical application of glucose transporter 4 is given in box 8.3. Absorption of other monosaccharides, glucose and galactose, are absorbed by the same transporter, SGLUT. It is an energy-dependent process, against a concentration gradient, and therefore absorption is almost complete from the intestine. Fructose is absorbed by GLUT5 transporter, Fig. 8.5. Other monosaccharides are absorbed by carrier-mediated facilitated transport. If absorption is not complete, the remaining molecules are fermented by intestinal bacteria.